Good afternoon, everybody. Thanks, everyone, for coming out on this Sunday when you have lots of other things to do, shopping, whatnot, family stuff. But I hope it'll be worth your while, and I hope you will learn a little something new, because in this world of MS, the new keeps on coming. I mean, it's incredibly exciting, and even before I get into my talk, I want to just tell you that I was recently reading that the Academy of Neurology, which is the umbrella organization of neurologists across the United States, has put out a position statement on treatment for MS, in which they are presenting the fact that we have these various therapies now available called disease-modifying therapies, that all patients should have the option of being on any therapy that is considered appropriate for them per the provider and per the patient's individual needs. And so I'm very excited that the Academy has stepped up to the plate to sort of push the issue with insurers as we meet more and more obstacles against prescribing these very expensive new drugs. And so the expense issue is also in their statement and ways of dealing with that. By the end of my talk today, I'm going to be presenting to you what are some of the obstacles for figuring out the right treatment for every patient. And so these obstacles, these challenges are what is going to lead us into a discussion of clinical trial work and the work that yet needs to be done. So that said, I mean, I was just reading about that this morning, and I'm very excited about the Academy's taking that step forward. Okay, so believe you me, this talk is not broad enough to cover all that's new in the field or all that's coming. I wanted to give you a sample, and much of this came out of a very big international meeting that took place this past fall in Boston, referred to as ECTRIMS. That is a meeting of European and American investigators, neurologists, clinicians, nurses, providers of all kinds who have an interest in and a focus on multiple sclerosis. And they met in Boston for a 3-day meeting, I believe it was, in which enormous amounts of data about new studies, new research, and new treatments were presented. And so it's impossible to give it all to you. I wish I could. I wish I could have absorbed all of it myself. But I've picked and chosen some things that I think are clinically pertinent for us to address today, and that may help you all in your understanding of what lies in the future for this disease. So I love these quotes. This is from Stephen Krieger, who is an MSologist, that's how we sometimes call ourselves, at Mount Sinai in New York. And he wrote this in an article in 2011, and I think it's still quite pertinent today. He says, since the introduction of beta interferon beta 1b in the early 1990s, multiple sclerosis has morphed from being a virtually untreatable disease to arguably the most dynamic area of new treatment methodologies and applied research in all of neurology. What does he mean? Means we have broken through the barrier, and it is an exciting time with a lot of movement forward. And then, in almost the next statement, he says, aha, but there's a problem. What's the problem? There are challenges that remain, right? And those challenges are as follows. Despite an expanding armamentarium of immunomodulatory medications that have demonstrated significant effects on relapsing disease activity, more effective and tolerable therapies are still needed for MS patients. So we've begun the work, but the work is not yet done. That's what he's saying. Okay, for that was my inspiration. Let's move forward with what we're going to talk about. So what is it that represents an obstacle to effective therapy nowadays in MS? Several. All of the MS drugs appear to only have partial efficacy in blocking relapses or blocking disease progression. There is no single drug we can tell you when you come into the office, you take this drug and you're done with MS. Would that it were so, but it is not, okay? Next challenge, adherence rates. I can give you a drug, but you're the one has to take it, right? So adherence rates for patients with MS have been less than ideal. Why is that? Is it because MS patients are more resistant to taking a medicine than other types of patients? No, they have the same challenges as any patient with a chronic disease. They object sometimes to the mode of administration, particularly when all we had were injections. They object to side effects. Okay, if anybody's had some terrible GI side effect, or if anybody's hair has fallen out, or if anybody has had worse MS effects because of a drug they were taking, if the treatment is worse than the disease, you're not going to stay with the treatment, right? And then additionally, in the MS world, we have some really worrisome toxicities. And by toxicity, we mean a therapy that, albeit it's helpful for the MS, but oh yeah, it could injure another organ system. And we don't want that for our MS patients. Our MS patients are often young and middle-aged adults. I see a very good selection right here of young and middle-aged adults, and they are not in a position to tolerate other organ system injury by virtue of their MS treatment. So that represents a challenge. Here's another challenge we have. Once we give you a treatment, we're not really sure how to measure whether it's working. Measuring treatment response in individual patients is not the same as measuring a population response in a clinical trial. We have 200 patients in this group and 200 patients in that group. We get 50 relapses in this group and only 10 in that group. That group wins, their drug is better. Is it better for everybody in that group? No, there were 10 people who had relapses. So it is very difficult to extrapolate from the clinical trial to the individual and say this particular drug is going to work for you. We don't have good measurements to tell us that. We don't have good markers that tell me patient A will do great on drug X. I don't know that. So what do? We keep examining the patient. We keep doing MRI scans. We keep measuring what we can measure when you're on a therapy, in the hopes that, in fact, we have picked the right therapy for you. And it turns out, sadly, that the effectiveness of each one of our therapies, of any therapy, is highly variable and unpredictable, since we don't have the right markers to tell us who's going to respond to what. We have one patient who comes in one day and says, "Doc, you gave me the greatest drug ever. It's now 2 years. I never feel like I have MS anymore." And I take that and I pat myself on the back. And then the next patient walks in and says, "Doc, that medicine you gave me a year ago, it's doing nothing for me. I'm having relapses. I feel worse than ever. The side effects are terrible." It's the same drug. So then I take the pat on the back and I throw it away. Because really, I don't have ways of predicting the differences in responses of those two patients. And that's a very sad thing and a very frustrating thing for the patients particularly, but also for the treating docs. Okay, so here's our landscape. These are the medicines we're working with, right? You recognize most of them. I gave you their generic name, their scientific name, and I gave you their trade name, okay? And almost all of them are in constant and active usage, with the exception perhaps of mitoxantrone, which has fallen out of favor. Why? Because of significant toxicity that was greater than what was predicted when it came to market. Those toxicities of novantrone, and believe me, if any of you here has been on novantrone, many people thought that was a great drug, myself included. It was incredibly, phenomenally effective, but it also caused heart muscle damage and it caused secondary cancer. So not a great set of toxicities for healthy middle-aged adults. So despite these therapeutic choices, we have these treatment challenges. To date, we have additionally no effective treatment for people with primary progressive MS and no proven treatment that works well in secondary progressive MS, when people have segued from the relapsing phase to the progressive phase. And again, in early relapsing remitting MS, we lack predictors about the disease that tell us who's going to have bad disease. That would be really helpful, right? If we knew at the very get-go who had a worse form of relapsing disease, we'd know who to throw the book at. Obviously, we would use more aggressive therapies or therapies with some potential toxicity if I had that omniscience to know who's going to do badly if I don't do this. We don't have those predictors yet either, so those are challenges. Other challenges, we lack biomarkers. What's a biomarker? That's something that I can measure in you. Blood tests, for example, or something on your skin that tells me you're turning purple. I know that's bad, right? A biomarker is something I can see or measure that reflects on the disease itself or reflects on what the best treatment for the disease would be. So for example, a biomarker like a neutralizing antibody against interferon is a blood test we can do. If you have high antibodies, it might mean that your interferon is no longer going to work for you because those antibodies neutralize it. That's a biomarker. A biomarker, if I test you for something called JC virus and you're positive, then that's a biomarker that tells me if I put you on the drug natalizumab, also known as Tysabri, you may have a small but real risk of getting a disease called PML, which is an infection of the brain, because you have that virus in your body. That's a biomarker. So yes, we are using biomarkers in a way to guide treatment. What's our biggest biomarker? MRI scans. We use that as the marker of what your disease is doing. Is it wholly accurate? No. Are we able to measure everything that's pertinent on a regular basis? Not really, because we know some of the pertinent things like brain atrophy or spinal cord atrophy are not routinely measured in MRIs. So we are lacking some of the best biomarkers to follow patients with the disease. So a good biomarker would be a biomarker that would help us select the exact right drug for the right patient. If I had a biomarker or a blood test that told me you would definitely be a responder to interferon, the game's over. I just put you on interferon and I'm done. That doesn't exist yet. We also don't have good biomarkers to tell us when treatment is failing. So one of the things we do is get an MRI scan, right? If we see new lesions, new MS plaques, or if the dye is being picked up by the MS plaques to suggest they're active, boom, we have a biomarker that tells us treatment's failing. Treatment's failing. That's really useful. But we have patients who are clinically failing and there is no biomarker on the scan to tell us why. We know they're getting worse and yet there's no marker, okay? So defining and knowing who's got breakthrough disease, who's actually getting worse, is still an art, not a science in MS management. And then once again, we would love to have a biomarker that would tell us who's got the worst disease. Tell it to us early so we can early be aggressive in our management. And maybe take more risk. Then I would have an argument for that patient. If I knew that patient was headed down the wrong path, then I can say, I need you to take this risk because you're going to have bad disease. And the risk of having bad MS is also a risk, right? There's risk of treatment, but there's risk of the disease wrecking you. So we want to weigh those two risks. I need better biomarkers to be able to do that. I don't have them yet. And so what I have here is a slide that lists sort of the topics that I'm going to address in the rest of this talk. Each one will be highlighted as I get to it. So, what about a new approach to acute relapse? Well, oral corticosteroids. What do I mean by that? Well, you might say, hey, we've been taking oral prednisone for years. Yes, we have been using low doses of oral prednisone to treat relapses for decades. More effectively, we use high doses of intravenous corticosteroid called IV Medrol, right? Many people have had IV Medrol at some point in the course of their disease to treat an acute relapse. Well, IV Medrol is a little cumbersome because it's IV, because it's expensive, because the IV keeps falling out, it keeps failing, you have to call the home health nurse. It's a pain in the butt, right? And it's expensive. So in Europe, they did a clinical trial of high-dose oral to match high-dose IV and see if they worked as well. And so the structure of this trial is what is on this slide for you. It was done in France, and what they were comparing was 1,000 milligrams of IV methylprednisolone to an exact comparable dose of 1,000 milligrams of oral methylprednisolone, okay? The design of the trial was called a non-inferiority design. They weren't trying to prove that one was better than the other. What they needed to prove was that they were equally effective, okay? That was the objective. The primary endpoint then was a score of disability or function called the MS functional score. That was the scoring system they used. And they were looking at patients at day number 28 as the primary endpoint. 1 month after your relapse, did you improve as well if you were on the IV version versus the oral version? That's how the study was set up. Patients in the study were the usual ages, middle-aged patients with MS. EDSS, the estimated disability status score at the study start, and you started the study when you were having a relapse. So the study start score was average 3.4. That's on a scale of 0 to 10. The EDSS score begins with 0. That's a person is normal if you have a score of 0 on that scale. If you have a score of 10, you're deceased. Usually, patients in the first 10 years of their disease are often in a range of 0 to 4, and we like to keep them there. We don't want them to go beyond 4 because beyond 4 is when you have ambulation difficulties. So at the time of entry into the study, patients on average were 3.4. At the study conclusion, meaning at 1 month, patients in both treatment groups had come back down to a score of 2.1. So both groups improved, and they improved on average to the same level, right? With either IV or oral steroids. They also looked as a secondary endpoint at how many relapses occurred after that, because we do know that giving people IV Medrol seems to hold off the next relapse. So the other important point here was, if I give somebody high-dose oral, will they have another relapse more likely 3 months later than if I were to have given them the IV form? And it turned out that the number of repeat relapses at 6 months out was the same in both groups. And finally, is it more toxic to give people oral high-dose steroids? We always worry about belly aches, gastritis, ulcers, etc. from steroids. Turned out that the safety of the high-dose oral Medrol was equivalent, with no excess and GI upset with the oral version. So, in effect, we now have a new oral option for acute relapses. Based on that study, many clinicians across the United States, and there have been other oral studies done prior to this, but this was probably the best done trial. We can now offer patients an oral alternative to manage acute relapse requiring steroids. Oral therapy is ideal for people who have poor IV access. Oral therapy may be preferable to you if you've got a busy work or school schedule and you don't want to be laying around with an IV in your arm. Oral therapy, in fact, is much less expensive, which is one of the motivators for having done the trial. So that was another little boost for us to have this alternative for treating acute relapses. We hate to have acute relapses in our patients. We'd rather our disease-modifying therapy be blocking them, but nevertheless, it's nice to know we have this alternative option. We've had several patients in our center take the oral steroids. Some say, great, thank you very much. Others say, that's the worst tasting stuff I ever had. Give me back my IV. Okay, so it's different for different people. Next topic, new versions of old drugs. We're going to talk about 2 new drugs, new old drugs, if you will. Copaxone, the 40 milligram formulation, and Plegridy, both of which have come to market in the last year or so. What was the data on Copaxone 40 milligram? This new old alternative. Everybody goes, oh, sigh, we know about Copaxone, it's a needle, it's a daily needle. Well, the pharmaceutical company knew that was an obstacle. So they decided to look at a higher dose taken less frequently. 40 milligram Copaxone taken 3 times a week versus placebo in a large, randomized, blinded trial. What happened? There was a 34% reduction in the relapse rate, very much like what the data was in the original 20 milligram Copaxone when that was studied many, many, many moons ago. There was also a 45% reduction in new gadolinium-enhancing lesions and a 35% reduction in new or enlarging T2 lesions. The T2 lesions are the white spots, right? The enhancing lesions are the spots that are picking up dye, which means they're currently active. So in clinical trials of relapsing MS, we always look at those MRI parameters as important data that tells us whether the therapy is in fact having an impact. And here we see classic reductions in the MRI markers as well as a reduction in the clinical activity, and that's what brought the new formulation of Copaxone to market. At the same time as they did the major trial, the GALA trial it was called, they did a study called the Glacier trial in which they compared 40 versus 20, not for effectiveness, but it was compared in a much smaller trial for tolerability. So the important thing to know was it more tolerable, in fact, to take a 3-time-a-week shot, or was that shot somehow less tolerable? And it turned out there were fewer injection site reactions because, of course, there were fewer injections for the 40 milligram group, and there was no excess in other side effects. And so this really was a new convenient drug that was brought to market. Okay, what about Plegridy? That's the new version of interferon, beta interferon 1A. So it's a newly improved interferon that's given less frequently than any other interferon on the market. And remember, we've got beta seron, which was the classic first interferon, that's interferon beta 1B. Then we have two versions, other versions of interferon 1A. There's Avonex, taken once weekly intramuscularly, and Rebif, taken 3 times weekly under the skin. So those were our menu, if you will, of interferons previously, and we know that interferons are very effective for MS. Well, Plegridy was a new version of interferon 1A given sub-Q. So they based it on Avonex. They took the Avonex molecule and they changed it. They added something called pegylation that prevents the molecule being cleared from the system as quickly, and so you have drug that lingers. And as a result, you can deliver it less frequently. So it's a twice-a-month injection, which, you know, I mean, that's getting down to bare minimum of injection, right? The advanced trial was the clinical trial used for Plegridy. It was phase 3 double-blind. That means people were either on placebo or they were on the real McCoy, but everybody was getting shots. And in fact, they did a once-a-month shot and a twice-a-month shot, and then placebo, because they were looking at how infrequently could this drug be given. And it turned out that the twice-a-month shot worked the best by virtue of these parameters. It reduced relapse rates by 36% over the placebo group, and it reduced disability progression during the trial by 38%. I want to tell you a little bit about disability progression so we don't get sort of blindsided a little bit here. The one thing we want in MS is to have nobody's disability progress, right? You know, if I were to tell you you can have 2 relapses a year, but on this drug your disability will never progress, you would say, okay, I'll take the relapses if I know I'm going to be okay, right? In clinical trials that only last a year or two, when we measure disability progression, we're measuring a change in score, and with the EDSS score, it means if you're getting worse, the score goes up. A change in score that goes up a point that stays that way for at least 3 months. That's disability in the trials. Is that disability in the real world? No, because some people will have a relapse their score gets worse for 6 months, and then they get better again. So that is not necessarily equivalent to the long-term progression of disability that we're all worried about. The long-term progression of disability is when I have somebody who had attacks 5 years ago and they have never, ever recovered. That's fixed disability, right? That's never going to get better. We don't measure that very well in trials. So when they say sustained disability progression in a trial, it's a little bit of a fib as far as linking that to long-term disability. But it's the best we got because trials can only last 2 years. They're very expensive to maintain and mount, and they're difficult to get through. It takes a lot of people and a lot of participants, even when they're only 2 years old, to make a trial go 5 or 10 years to actually prove changes in disability progression over the long term is virtually unaffordable for sponsors of trials. So it's difficult. But within this Plegridy trial, there was in fact a limitation on disability progression in the treatment group versus the placebo group in the short term. And then the MRI data was very good. There was a 67% decrease in new or enlarging white spots, the plaques, in the interferon-treated group. So what's the motivation to have new stuff on old drugs? Well, clearly, there were some objectives that were met here. The pharma would like to improve patient comfort and convenience because this is going to result in what? Improved adherence. Better patient compliance means the drug works better. If you don't take the drug, I guarantee you it doesn't work, right? So, you know, when people come in and tell me their Copaxone's not working and boy, they missed 5 shots this month, I gotta sit and worry, right? Over the long term, we see better efficacy when the patient is consistently on drug. Of course, there's another motivation, and that is that both of these companies, the original drugs, the Copaxone 20, was going to lose its patent, as will Avonex in due course. And so the motivation is also to have a new name brand, if you will, to maintain that brand on the market for a longer period of time. So there are mixed motivations here, but nevertheless, it's to the patient's benefit if they can get a more convenient drug. And I must say that I do have a number of patients that have moved on to these new products with great relief of injection frequency and with continued efficacy of a drug that worked for them. So if this drug works for you, why move on, right? Next topic. We're going from the old to the very new. I want to talk a little bit about a drug called Lemtrada, whose generic name is alemtuzumab. And that is a newly approved infusible drug. That means IV. That came to market just in recent months. So there was a lot of hoopla because initially the FDA denied approval of this drug, and there had to be more evidence brought back to the FDA, more discussion, more review, and then finally it did reach approval and is a new, very powerful agent on the market for treatment of relapsing forms of MS. What is this drug? It's a humanized monoclonal antibody. What does that mean? That means they take an antibody that's derived from a mouse and they humanize it by putting stuff on it that makes it look more like a human antibody. Why do they do that? So your body doesn't reject it. So you don't make antibodies against the drug. They want it to look native to the human body, okay? And it has a particular binding site which is on the surface of certain white blood cells at a marker called the CD52 marker. What happens when this drug binds to those cells? It eliminates them, knocks them off totally. So where is this marker found? It's found on very important cells that have a role in the immune misactivity, if you will, the misguided activity that we see in MS. So T lymphocytes, B lymphocytes, and also some other white blood cells carry this marker. So this marker marks them for death when you take Lemtrada, okay? It knocks them out. Now, we used to do this decades ago in a more potent way with chemotherapy. When we had nothing good to give patients with MS, we gave them bad stuff. We gave them Cytoxin, we gave them mitoxantrone, Novantron. Those drugs are chemotherapeutic and they kill off white blood cells. This is also depleting white blood cells, but it is much more selective. It's not quite as broad. But after a single initial course of treatment, which is 5 daily doses given IV of alemtuzumab, the B cells go away. Most of the B cell population goes away for 6 months. And the T cells are reduced in number for a year or longer. That's pretty potent stuff, okay? Then the plan is to repeat the dose of alemtuzumab once a year. Okay, so we do it again a year later. And then the second dosing schedule is actually only 3 days. Because of the potency and because they've already been treated once, people usually don't require more than a 3-day treatment at 1 year. And you cannot treat at a year out unless the counts have recovered to certain acceptable levels. So how was alemtuzumab brought to market? What were the trials about? There were 2 large trials, and in both of those trials, they were structured comparatively the same. In both trials, alemtuzumab was being compared to another active therapy, interferon 1A. And in this case, the attempt was not to prove equivalency of the 2 therapies, but to prove superiority of one therapy over another. And that takes a lot more patience, and that takes a lot more potency of the drug to reach a statistical level where one drug outperforms another drug. That's very much like stroke trials, where every time a new drug is brought onto the market for stroke, it has to beat what? Aspirin, right? Aspirin's a great, cheap anti-stroke agent. That's how they prove new drugs to the market for stroke. Similarly, that's what they wanted to prove in these 2 trials, and they did. Both studies showed a 50% reduction in relapses in the alemtuzumab group as compared to the interferon group. And in one of the 2 studies, not both, but in one of the 2 studies, they proved a difference in disability progression during the 2-year trial. So in only one of the 2 studies were they able to prove that alemtuzumab beat out interferon 1A rebif for disability progression, okay? There were lots of MRI measures, T2 lesions, new T1 holes, enhancements, etc. They all favored alemtuzumab over rebif. So that's really dramatic, and that's why the MS population and many investigators really wanted to get this drug to market, because clearly it had taken a step above and beyond a therapy that had long been accepted as a very effective therapy in the MS world. They looked at also people with certain forms of MS, and it turned out it was particularly effective, Lemtrada, in patients with highly active and aggressive disease. So people who came into the study who had more active lesions, who had more disability, were in fact more benefited by Lemtrada than by rebif, okay? And of course, it's a pretty convenient drug to take once a year. How about that? You're 5 days of IV and you're done for the year, right? So that was really, really impressive with this drug. And the fact that it outperformed a standard drug. But there are some disadvantages of this new drug, and they are not small. Infusion reactions. By infusion reactions, we mean like an allergic reaction, like a rash or chest pain or dropping your blood pressure or having shaking, chills, and fever. Those are typical allergic infusion reactions that, by the way, are seen with a lot of other chemotherapies and other drugs on the market, other antibody drugs for other autoimmune diseases. That's not unusual. But it's seen in up to 90% of patients. So we have to be very prepared when you administer this medication that you have effective therapy to treat the actual infusion reaction. The other thing seen in the alemtuzumab groups over the course of these 2 trials was there was a mild to moderate increase in infection rate of just routine infections, not opportunistic or serious or deadly infections, but nevertheless infections. And what was particularly worrisome and impressive is that a full third of patients who take alemtuzumab suffer failure of thyroid function. It shuts down their thyroid gland. And that means they have to go on and take thyroid medicine for the remainder of their lives. So this is a drug that has very high potency for the disease, for one autoimmune disease, but it causes another disease in a significant minority of patients taking it. In addition, other serious autoimmune diseases were noted to occur in a very small percentage of patients, probably less than 1% for all of these. But these are different autoimmune diseases. One of them is called ITP, immune thrombocytopenic purpura. That is a disease in which the platelets drop off. The platelets get destroyed. And the platelets are your blood clotting element, so that can cause bleeding. Autoimmune hemolytic anemia causes anemia by rupturing red blood cells. Goodpasture's disease causes injury to the lungs and the kidneys. And glomerulonephritis is an autoimmune inflammatory disease of the kidney alone. So these are not small potato diseases, if you will. This is not, good stuff to get, but it's in a very small percent. So you have to weigh the incredible efficacy of this drug, the potency of it, the fact that it puts people into remission who have very aggressive disease, on the other hand, its potential toxicity. And there is a stringent REMS, which is called Risk Evaluation and Mitigation Strategy. That's a cocktail of steps that the FDA mandates in order to use this drug. So the users, in our case, our staff at the MS Center have to be trained in its use, and we have to know all of the follow-up that's needed to be done to keep patients safe after they receive the drug, as well as the detailed protocol for administering the drug safely. So all of that is getting in place at our center and in other centers where this drug will be used. So that is a new drug, and that is a new effective agent in our armament, but with some concerns. Okay, moving on. What about some other things that you've heard in the background for years and years? What about estrogens? What about hormones for MS? What about anti-lipid agents? Everybody keeps talking about Lipitor for MS. What new things do we know about those agents? Estriol is the medication that has been trialed. It's the hormone that is thought to be the candidate effective hormone in third trimester pregnancy. We know the best drug we have for relapsing forms of MS is pregnancy. Well, it's the best in some ways, right? Okay. Not good for our male patients, not really accessible for them, not great for our patients who have already got a lot of babies, but it really reduces relapse risk, particularly in third trimester pregnancy. And estriol is the identified estrogen that seems to be the operational estrogen that causes that. So there was a recent large trial of estriol, which by the way is not on the market here as for hormone replacement therapy. It is not in the United States. I think it is in Europe, but they combined estriol with glatiramer acetate Copaxone and compared that to placebo plus Copaxone. And in the first year of that 2-year trial, there was a definite drop in the annualized relapse rate in the combined therapy. That looked very exciting until, unfortunately, by 2 years into the trial, the differences in the two arms were negligible, were not statistically significant anymore. So what looked like a great add-on for preventing relapses didn't hold by the second year. What else did we learn in that trial? MRI differences. That would have been the ticket, right? If we saw some MRI data differences between the two treatment groups, combination therapy versus Copaxone alone, but in fact, those MRI measurements were not different in the two groups. And this data was all presented at the meeting that I was at. Interestingly, but as a standalone phenomenon, I don't know how much that will contribute to getting estriol to the market, there was an effect on cognition in the first year of the trial with better outcomes for the estriol group on the PACEAT, okay? What's the PACEAT? The Paced Auditory Serial Addition Test. Has anybody ever taken that horrible test? Where you listen to a series of numbers. The recorder is going 1, 3, 5, 7, and you're supposed to add the current number and the one right before it. So after you hear 1 and 3, you say 4. But then they say 5 and you have to add 3, the last number, plus 5, and say 8. Don't go back to the one at the beginning. Anyway, my description of it is just as horrible as taking it. It's really awful, and patients hate it, but it is a way of measuring cognitive accuracy, and it's used in a lot of clinical trials, right? And it turned out that taking estriol in the trial seemed to give you an edge for performance on the PACEAT, at least in the first year of the trial. So there are some cognitive advantages perhaps brought by estriol, but the jury is still out on whether this is going to be any kind of marketed addition to our armament. What about Lipitor, simvastatin? There have been a number of trials of Lipitor over the years. There was one small combination trial I heard presented years ago where they used Lipitor with beta-seron versus beta-seron alone. It was very small numbers, but the people on the combination did worse. But there have been other trials in which it looked like Lipitor had a beneficial effect. In this particular trial that got presented, Lipitor 80 milligrams, which is pretty high dose, was given to a population of who? Secondary progressive MS patients. Patients who are neglected, who are understudied, and have not really gotten the opportunity for an effective treatment, right? And so what they did, it was a phase 2 trial. Jessica, who's going to follow me, Jessica Schulte, who's going to talk about the conduct of trials, she's going to define these different phases for you. But let's say just here that it means it's a small preliminary trial. The object is not to prove effectiveness of Lipitor in a big way, but to see if there's a trend that make it worthwhile to study it. So in this phase 2 trial of patients with secondary progressive MS who were either on the Lipitor 80 milligrams or not, there was a difference in the progression of brain atrophy on the MRI. Brain atrophy is really thought to be an important sensitive measurement for patients with progressive forms of the disease, a measurement that we don't do very well in usual MRI centers in clinical settings like the hospitals. We don't yet do that very well, but in research studies they do. So they made these measurements and it favored the Lipitor as preventing atrophy progression, which means in progressive disease we think one of the reasons for progression is loss of gray matter. And so if you show that there's less atrophy with this drug, maybe it's having a protective effect on the gray matter. At any rate, finding support moving forward with a larger phase 3 trial in the hopes of demonstrating some clinical correlates. That is, can we show that the patient's progression clinically is less if we give them Lipitor? Not only is their MRI better, but are they going to be doing better? And so that'll be the next stage of development with Lipitor. Okay, next topic. The one question we hear all the time in the office, all of us, Mary Kay probably more than anybody, is what can I do to prevent getting worse as far as my diet, as far as my exercise? Well, patients don't always ask about exercise because they don't really want to hear the answer, but is there anything I can do? What's the supplement I can take that's going to make me better? What can I buy at GNC or the health food section of the store that's going to make me better? And so the important data that came out as far as environmental influences are these three that I'm going to mention: salt, smoking, and sun. Okay, the 3 S's. So, sun, vitamin D. What do we know now about vitamin D? We know that low vitamin D levels is a firmly established risk factor for getting MS. So we don't want people to be vitamin D deficient. And I do tell patients, you want to improve the risks for your children? Tell them to take vitamin D. Get their vitamin D levels up. And as Mary Kay loves to say in her talk, to really get enough vitamin D from the sun, you have to have 85% of your body exposed for 15 minutes a day. And she, like I say, we never expose 85% of our body to anything or anyone. So therefore, we're never going to make it that way, right? So you're going to need to take vitamin D. Vitamin D administration has also been proven in the animal model of MS to decrease the severity of the attacks and the progression of the disease. In the guinea pigs and the mice, well, that's real nice, but people aren't guinea pigs and mice. Now, however, there have been a number of, albeit small, human trials in which they looked at vitamin D as an add-on supplement or health trials over years where they looked at who got MS, and both of those suggest that vitamin D replenishing not only decreases risk for getting MS, but modifies severity and progression of MS once you have it. Now, the data for that is still very shaky, but more trials are going to follow. And so for now, it's a harmless thing to supplement and normalize one's vitamin D levels. So we are recommending that to patients. What's the salt story? That's a new story, okay? And over the years, I've heard the salt's bad for high blood pressure. Then they said, no, it's not really bad for high blood pressure. Then they said, well, added salt is okay, but it's the salt in the processed foods. We keep getting mixed messages of what to do with salt. But what they did was they looked at a high salt diet in the animal model. EAE is the animal model, experimental allergic encephalomyelitis. They make up an animal model of MS because no other species but humans gets MS. So this is an artificial way of creating MS in a mouse, okay? And what happens is a high salt environment in the diet of the animal, a high salt environment turns out to be an inducer of activation of T cells called Th17 cells. That is one form of T cell that turns out to be a bad guy cell in a lot of people with MS, is that they have too much activity of the Th17 cells. So these cells are potent producers of what are known as pro-inflammatory cytokines. That is, proteins that message the immune system to get going and start attacking the nervous system. So they looked at that in the animal and they said, well, maybe that is operative in people. And in fact, in a 2-year observational study of a group of MS patients, there was a much higher relapse rate and a greater chance of new MRI-T2 lesions in the patients who had medium or high salt intake in their diet compared to the patients with low salt intake. That's just one study, guys, but it's of consequence perhaps. And so that's something you can do something about. You can alter your salt intake by virtue of looking at salt content on foods that you buy, avoiding processed foods. Everything has salt. Oh my God, everything has salt. But then I watch, what's that French chef? Pepin. I love him. And every dish he makes on TV, it doesn't matter what it is, and a little salt, and he keeps a bowl of salt. He's just throwing in the salt, and I'm sure it all tastes great. But anyway, that is a dietary modification. People with MS have been asking us for decades, how can I modify my diet and stay healthier? Well, guys, that's a modification. Okay, what I don't have a slide on is the smoking. Forgot to include a separate slide, but there have been now a number of observational and health trials that demonstrate that smoking is a risk factor for getting MS. But indeed, smoking in patients with progressive forms of the disease makes the progression worse. Okay, so you'll have to take my word for it. I forgot to make a slide to show you some of that data, but it's true. And so like everything else, smoking is bad for you. There's just another good reason on the list. Just check off another one. When you have MS, smoking is bad for your MS. Okay, so be sure to take adequate vitamin D, consider lowering the salt in your diet, and for heaven's sake, stop smoking. Here are some of the new things in the pipeline. For relapsing MS, 2 more antibodies, ocrelizumab and daclizumab. For progressive MS, 2 other agents, laquinimod and ibudilast, and to treat both relapse and to enhance myelin repair, something called the anti-LINGO molecule. And data about all of these was presented at the meeting. So what are we talking about? New agents in the pipeline, ocrelizumab. We don't have a trade name yet for this. Whenever you see MAB, MAB is a mouse antibody. So Zumab is a humanized mouse antibody. Natalizumab is Tysabri. Alemtuzumab is the drug I talked about earlier, Lemtrada, that's come to market. Now this is a new one that's in trials, including trials that we're involved in at our center. It is a B-cell depleting agent that's being investigated both for relapsing and progressive forms of MS. It binds to a different receptor, a receptor that's unique to B cells in certain phases of B cell development. So it doesn't affect the most immature B cells, the stem cells in the bone marrow, so you keep making B cells. It also doesn't affect the most mature B cells that make it to maturation, the ones that make you antibodies in your body. We all need B cells because they make us antibodies. It affects the B cell that goes in between in the evolution of B cells. And it's very similar, this drug, to a drug that's already on the market called rituximab or Rituxan that was brought to the market to treat what? B cell cancers, B cell leukemias. And so it knocks out B cells that are abnormal and reproducing abnormally. And so they took that drug, they modified it, and made ocrelizumab to treat MS by selecting out some of the bad B cells in the MS immune pathway, if you will. So it's very similar in structure to the Rituxan. There was a phase 2 study, phase 2 small preliminary study that showed a reduction in relapse rate and also in all the MRI parameters when they compared this drug, ocrelizumab, to placebo, as well as when they compared it to another active agent, interferon 1A IM, which we know to be Avonex. Okay, so those comparisons did show a benefit in small trials. Now the 2 large phase 3 trials are ongoing. They're referred to as OPERA-1 and OPERA-2 studies, and that's for relapsing disease. But there's a third trial called Oratorio, a trial of this same medication for people with primary progressive MS. Also a very large trial. We're talking about 1,000 patients in each trial. And those trials, the ones for relapsing remitting, probably we're going to get results within, what, Jess, 6 months? They will have their data analysis, so hopefully it'll be announced, if not this spring meetings of the AAN, in the fall meetings for the same meeting that I went to last year for Actrams. Okay, so that's what's going on with ocrelizumab. It's given IV every 6 months, so convenient. The B cell depletion repopulates in that time, so it's not quite as long a lasting effect as what we see with Lemtrada. There is no effect, as I said, on the plasma cells that have to produce antibody, and so far, thank God, no limiting toxicity on other organ systems has yet been seen. Another antibody called daclizumab, they completed their trials, which were presented, and I'm surprised they haven't come to market yet, but I think the FDA is considering this drug currently. This is a monoclonal antibody that blocks a different receptor that's found on some types of T cells, called the IL-2 receptor. IL-2 is interleukin-2, and what this does is it alters the balance of different T cell types in the immune system of patients who are treated. And there is a set of cells called the CD56 cell, or also known as natural killer cells. It's actually a good cell in that it has regulatory effects on the immune system. And CD56 is thought to be one of those cells that exerts a suppressive effect on bad behavior in MS, bad immune system behavior. This particular drug is given once a month, sub-Q. So now we've got a drug that's a shot, but only once a month. That's a very good thing for convenience. The phase 3 trial, I'm sorry this print is a little small, compared it to interferon 1A intramuscular, that is Avonex. The primary outcome showed a 45% decrease in relapse rate with the new drug compared to Avonex. The MRI outcomes showed decreases in new and enlarging lesions, 54% drop in that, a 65% drop in gadolinium enhancements when comparing those two drug types. Skin rash, injection reactions, and increased liver function tests. We've heard that before with MS drugs. Those were the side effects, okay? But those were the only significant side effects. There were no serious infections or other toxicities seen. This drug looks like a keeper. We're waiting for approval. This is a new drug for MS. What about this drug, laquinimod? Maybe some of you have heard about laquinimod before. There was a parent drug to laquinimod years and years ago called linamide that was studied in trials, and sadly, it made it all the way to large phase 3 trials. It was thought to be a great drug, and it turned out to have untenable cardiac effects. There was an increase in heart attack in the treatment group. So that's how far a drug can come. It was in the final phase trials, and they had to throw it out. But they took that molecule and they reconformed it. They looked at various entities on the molecule, and they redesigned a new molecule that they thought would not have cardiac toxicity, and it turned out to be so, and that drug is called liquitamod. And there was this long road to developing this liquitamod, and then they finally got it to a big phase 3 trial for relapsing remitting MS, and lo and behold, it didn't do what they wanted. There was a disconnection. What was the disconnection? It had effects on disability progression in the study, but it didn't have enough efficacy on relapses. So you can't get a drug that doesn't affect relapses to market for relapsing forms of MS. That ain't gonna go. So they have now looked at this drug in a new way, saying maybe this is the right drug for progressive forms of MS. Because they have found that this drug actually gets into the central nervous system. It actually induces a protein called BDNF, brain-derived neurotrophic factor, which is thought to protect nerve cells from degeneration. So in fact, liquitamod may prove to be a ticket for progressive forms of disease, and the new trials are already underway for this drug with a new objective, which is to affect progression. Ibudilast is also a drug that's being trialed in secondary and primary progressive disease. There's a center at WashU that is looking at this drug. It's anti-inflammatory, but it prevented both demyelination as well as exerting neuroprotective effects when they studied it in the animal model of MS. The outcomes in the trial that are currently underway are only MRI outcomes. It's phase 2, it's an earlier phase trial. Only if the MRI data comes out positive will they then move on into the large phase 3 trial. So that's on the way, we hope. There is also this other agent called the LINGO-1 protein. The LINGO-1 protein blocks the maturing of myelin-making cells. Myelin-making cells are called oligodendrocytes. Those are the myelin-making cells in the central nervous system. They go from a precursor cell, an immature cell, to becoming a mature myelin-making cell with certain signals, okay? One of those signals is called the LINGO-1 protein. When LINGO-1 is active, the cell does not mature. So the drug that they're studying is an anti-LINGO-1. That is, it's an antibody that blocks LINGO-1 and results in premature or immature myelin-making cells becoming mature myelin-making cells. So in effect, you're speeding up the number and activity of myelin production. Therefore, then you will get more myelin repair, because there's always remyelination going on in MS, but we lack enough cells to do it. So remyelination and myelin repair were both enhanced in the animal model of MS when the animal was given this drug. So right now, there are 2 trials going with this anti-LINGO molecule. One is in relapsing remitting patients. The other is in patients with acute optic neuritis to see if we can repair the optic nerve and have a better outcome of optic neuritis for the long term. So let me conclude with just a few remarks about strategy, because I know that's a lot of data, and that's why I wanted you to have the slides. You can read all about it and, email me questions if things come up. But what do we do now with MS patients? Here's what we do now. We sequentially give them monotherapy. What do you mean by that? Well, the tradition thus far is we choose a drug, we give you a drug, and when that drug doesn't work, we go to the next drug, okay? And the other thing we do is we escalate treatment. We start with safe, non-toxic, long-proven drugs, okay? And then if you don't do well, then we go to a riskier, not so safe, newer drug, okay, because you're having breakthrough disease. That's kind of the way we do now. What's wrong with the way we do now? Well, apparently plenty, because it doesn't always work. We have limited definitions of knowing who's failing treatment. I already told you that. The MRI is not wholly accurate in telling me who's failing treatment. We have very few head-to-head trials. So when I select drug 1 and then I go to drug 2, I don't really have a trial that proves that drug 2 is better than drug 1. I may know that it's riskier. It may have more side effects, but that doesn't necessarily mean it's stronger. We believe some of our drugs are stronger than others, but belief is sort of like, mythology. You can believe it, but that's not data. And we have poorly defined measures, as I've said, that confirm when patients are failing. So how can we change our strategy? What can we do different that will make it a more logical, more scientific approach? Well, we may want to, here's some options. We might want to consider using the aggressive and risky therapy for a short time early in the course of disease in everybody as a form of induction. Let's shut this down early, powerfully, in everybody who has MS, who comes with a first attack. And maybe then, after we see that we've got them into a good remission, then we switch to something that's a little safer or lighter weight. Maybe that should be the way we do things. Or more logically, wouldn't it be if we could choose a drug based on how that drug works, on the mechanism of action, and then switch patients from one mechanism of action to a different one based on how they're doing, or even better, based on the immunologic subset of that patient. If I knew you to be that patient who has too many Th17 cells, then maybe I'd want to give you the drug that particularly knocks out Th17 cells. That would be more logical, right? That's one new approach we might be taking once we get scientific data that helps us. Or the future may be combination therapy. Look at the chemotherapy world. Nobody gets one drug for cancer. They get 3. They get one that kills the cells. They get one that prevents the blood vessels from getting to the tumor. They get one that enhances anti-inflammatory effects. Then they get a bunch of drugs to tolerate the first 3 drugs, right? So nobody gets away with one drug for cancer. I'm not saying cancer is the ideal treatment, but they have some really great successes. We need that kind of success. And so it may be that combination therapy will be the answer, but we need more combination trials. The estriol trial was very worthwhile to tell us some data about combining estriol. We need that for many, many other agents. So advancing the science of this MS therapeutics world really means we're going to have more basic science investigation into what makes this disease go, what's the pathology, what's the immunology, what are the differences between subsets of patients. We need very large controlled trials that unfortunately are very expensive that compare the existing treatments to the new ones as they come out. So we know, is this new drug really better? And we need the participation of patients with MS. And it is very time demanding to be in an MS trial. And it is risk-taking to be in any trial. But that is absolutely essential, as you saw today, for any of these therapies to make it to market.